Okay, so welcome to this lecture extra that I'm going to give you about the seaweed physiology lectures from the Marine Physiology and Behaviour module OSX-2002. So this is really just following on from what I went through in the lecture on seaweed reproduction and life history. Okay. So essentially, we focused really on how do seaweeds have sex, which is the first question. And some species, like fucoids, have specialized appendages known as receptacles. And within these receptacles, they have these pores, which are also known as conceptacles, where they store the sperm and the eggs. Many fucoids are dioecious, so that they have very distinct males and females. They are morphologically identical, but their sex organs are different. And in this slide here, you can see that the males have these orangey coloration, orangey conceptacles, whereas the females have green ones, and they're a bit darker. And you'll be able to see the difference once they become reproductively capable. Now, fucoids have flagellated sperm, so the sperm can swim, and the female eggs release a pheromone that attracts them. This is an important adaptation for seaweeds, because once they release their eggs and sperm into the water column simultaneously, it gets diluted. This mass, this basically big cloud of egg and sperm gets diluted very quickly by the seawater around the host alga and the algal stand. So the ability of the sperm to swim and to be attracted directly to the eggs increases fertilization success, making this a successful route to fertilization. Once fertilized, the egg and sperm combination turns into a zygote. That zygote is just a fertilized egg, essentially. It then undergoes a process called polarity, where the cell will then begin to know up and down through the diffusion of different elements, and it will form a zygote at the bottom of the cell, a rhizoid, at the bottom of the zygote. And that rhizoid will turn into a holdfast. For alga, for plants, it may be a root system. And this picture here shows some fucoid zygotes and the initial formation of the rhizoid. That, over a period of several weeks, will turn into a germling. And a germling is just another name for a baby seaweed. Okay? A germling is just a baby seaweed that eventually will grow into an adult alga. And then that cycle will then repeat around. Now, red seaweeds do it slightly differently because their sperm are non-motile, so they can't move. So they have developed different adaptations. That usually is basically a carposporophyte phase here. This carposporophyte phase is just on a female gametophyte, which is a reproductive female, and that carposporophyte will stay on that female. The male will release sperm. That sperm will end up inside the sporophyte, the carposporophyte, and then that will then produce a carpospore, which will be a single spore that will grow into a tetrasporophyte, which is very morphologically similar to the gametophyte. And that carpospore will turn into 4 tetrasporophytes. So you've got basically a quadrupling of reproductive success from a single fertilization by the introduction of that tetrasporophyte phase. And that is in occurrence here with an example of Chondrus crispus, but nowhere most red algae undergo such a stage. Some red seaweeds have male and female halves to them. So for example here we have Porphyra purpurea, also known as purple lava, very common and commercially important seaweed. And we can see in this picture here, we can see this zipper type structure. We have a male half and a female half, and once the male half has released its sperm it then sheds that material because it no longer needs it. And the eggs are retained in the female to produce carpet spores and so on. Now this one here, red seaweed Palmaria palmata, is a common red seaweed found on the shores of British Isles, and it's usually found on stipes of certain laminarian species, the large brown kelps. And this representation here shows this stalk sticking out of a female gametophyte. Okay, really it's macroscopic gametophyte, so you can't see it, 0.1 millimeters if you see. And this is called a trichogyne. And this trichogyne is basically a feeler that will pick up sperm, and sperm will attach onto that, and then that will undergo fertilization of the female. Now, in Palmaria palmata, it's usually older males fertilizing younger females. So that sometimes that reproductive strategy will allow the male to grow and get ready to reproduce. So these life cycles can be condensed into this single slide that I've shown you here. Okay? Now, some species have what's known as a single morphological stage or phase. And here it's represented in this one, which is a haploid life cycle, where the individuals go through their life with half the amount of chromosomes that are required for sexual reproduction. In this one, we have full chromosome complement, aka diploid life cycle, and then when ready to reproduce, it will undergo meiosis, produce eggs and sperm, which will then fertilize to form a zygote. In the haploid life cycle, they spend most of their time as kind of like eggs and sperm, but really as spores, which will come together to reproduce and form a zygote, which will then undergo meiosis, and then that will grow into the individuals that go through their life cycle. Now, the diploid life cycle is commonly referred to fucus and sargassum, and some tropical green algae like Hanamida and Caulipa undergo this diploid life cycle. And it's quite a common mode. Basically, it spends all of its life as the adult that you see on the shore until the eggs and sperm are produced, and then they produce a zygote which grows into the adult. Some species undergo a two morphological stage process where you have a sporophyte and a gametophyte. For some species, these two stages you cannot tell the difference with the naked eye. That's known as isomorphic. In others, you can, in that the sporophyte might be a really large individual, but the gametophyte might be microscopic. And a good example of that is laminaria, where we have an extremely large kelp, which is the sporophyte, but the gametophyte, the actual thing that will reproduce eventually, is microscopic. We can't see it with the naked eye. Now, isomorphic strategies are basically identical morphologies in the two stages, and identical environmental requirements for most species, but they have different reproductive structures, and they may be haploid or diploid, depending on what's going on. The two stages. So at the cytological level, the cellular level, they are different, but at a morphological level, they are not. Heteromorphic, very different in the two stages. As mentioned previously, we talked a little bit about seaweed sex for the red seaweed. They undergo this tetrasporophyte, carposporophyte phase, which is an element of increasing the reproductive success of the individual. They too have isomorphic, as we saw that the tetrasporophyte and the gametophyte of Chondrus were morphologically similar, but cytologically distinct. And then there are many more kind of heteromorphic life stages, so they're very different in the way that they look. And we'll have a look at some of these in a minute. So nobody really knows why seaweeds have such complex life histories. However, they do suggest that there are some theories that are out there. So they think that the different life histories having maybe different kind of levels, different morphologies and so on, may alleviate pressure that's selecting for one part of the life cycle or the other. You don't want to reduce the reproductive success or the establishment success of your offspring, because that would go against sexual reproduction and what the whole point of it is for. And people, obviously, the act of meiosis introduces genetic variation and zygote formation into the population by getting traits from the parent female and the parent male. And Matt Dring suggests that these are just serendipitous evolutionary experiments that just seem to work and just have stuck, but nobody really knows why. So there's different ways of looking at it. Now, in my lecture, I told you a story about asexual reproduction and a particular species known as Sargassum muticum, which I've written there. Okay? Sargassum muticum is a Japanese seaweed which was transported into the Atlantic first via the Mediterranean on oysters, and it landed on the coast of Britain in 1973 on the south coast near the Isle of Wight. And since 1973 it has spread all the way up our coastline, forming large stands which are nuisances in marinas and sheltered bays and lagoons. And it's basically a problem invasive species. And the reason it spreads so quickly is because it undergoes a process called vegetative dispersal, where you have your adult alga, maybe a wave or something breaks off, or a grazer is nibbled away, breaks off a part of the adult alga. That survives for several weeks in drift. So it drifts along in the tides and currents until it might not hit some suitable habitat. It then settles, forms a new holdfast, and then that then essentially is a clone of the host of the old adult that it broke off from, will form a new reproductively successful, reproductively capable Sargassum. Sargassum can also asexually reproduce by self-fertilization, which is a whole other problem. That does mean that you only need one, it will spread dramatically. And that reproductive success has been put down, and that success of colonization and of the invasion has been put down to the vegetative dispersal and also the selfing. So Sargassum is an incredibly interesting species for looking at this kind of invasive dynamics, and I talk about that in later lectures. So here we have some representations simplistically from Gill and Foster, which I recommended as a good thing to have a look at, good paper, is that we have mature fucoid produces a conceptacle within the receptacle, eggs and sperm are extruded, they're fertilized to form a zygote, okay? In some cases you may have self-fertilization. These produce propagules, which then grow into an adult alga, okay? That's it. Easy peasy lemon squeezy, really. Really simple life cycle. More complex life cycle is the fact that kelts produce spores. So you have a very large adult mature sporophyte. This brown area on the picture is known as a sorus. That's where all the spores are retained. The spores are then extruded, and then they form a gametophyte. And the gametophyte is a sexual reproductive element, which then fertilized. And that zygote then produces into a sporophyte. So essentially here, we have the sporophyte stage in the life cycle here, which undergoes its meiosis, producing spores, which then spores then grow into gametophytes, which then produce eggs and sperm, which produces zygote, which then grows into a sporophyte, and the whole cycle reproduces again. Okay? So a spore will just grow into a germling or a gametophyte. A gametophyte needs to be fertilized in order to grow. Okay? We now look at more complex life cycle, which is the multistage life cycle of chondrous crispus. So, here we have a male and a female gametophyte. Females have this thing here called a carpospore. Okay, carposporophyte. That will receive the male gamete. Once fertilized, the carposporophyte will produce a carpospore, a single carpospore, which will then grow into a tetrasporophyte. But they will produce 4 of these carpospores. Single carpospore will grow into a tetrasporophyte, and each tetrasporophyte will produce 4 tetraspores. And these tetraspores will then grow into gametophytes. So there's a whole process here of producing a carpospore, and a tetrasporophyte basically takes a single carpospore and then creates 4 tetrasporophytes, each capable of producing male and female gametophytes through meiosis. So this whole process of reproduction essentially is about increasing the success of settlement by taking a single fertilized carposporophyte creates 4 tetraspores. So you can imagine just how many of those go through and grow, because remember, sperm in red seaweed are not movable. They do not move, they do not get attracted to, they just end up serendipitously where they need to be at the right time. Polarity, which is a filamentous red alga, undergoes some processes which are similar to the whole process and that cycle. Very similar. Carposporophyte, carpospores turn into tetrasporophyte, grow into germinating tetraspores, which then produce gametophytes again, and the whole process goes round and round and round and round. So really, really clear. So the next major thing that you need to know about is the ecological context of reproduction and how that whole thing works. And I look at the 3 stages of reproductive success, which I call settlement, attachment, establishment, based on work by Fletcher and Callow in 1992. Settlement is how you get a fertilized zygote growing into something. It must first hit a suitable habitat. So some zygotes can reject a substrate purely because it's not suitable for it. And ova is an example of that from the Callow and Callow work in 2006. You might need a bit of roughness to allow your zygote to get trapped or to settle in order to not get washed off by waves. And this is an example here by Trevor Norton and Sargassum muticum. But you also need, and quite commonly you'll find that canopies facilitate growth by enabling the formation of a biofilm, a microbial mat almost, of extra polymeric substances. The polymers are growing together. It's called EPS, together in a microscopic biofilm which has got silt, it's got microbes, it's got everything growing all over it. So basically, if you get trapped in that matrix, you're protected somewhat from being washed off by a wave. And that basically increases the success of settlement dramatically. But you must also think, so some species would absorb water onto their cells in order to increase their weight and sink down to the bed. Okay, but essentially, even though some zygotes, some spores, some gametes and so on, they can swim, but actually they're at the mercy of the currents and tides, which are far, far stronger than them. Once you've settled, you need to attach. Otherwise, you're going to get washed off. So, species like Astrophyllum medosum require several days of extreme calm for their little components to settle and to attach. It can take many days. So all you need is one wave. 90% of all of the settled gametes are just washed off. The zygotes are just washed off. Okay? Ova has a glue-type substance that it excretes that allows adhesion onto the bedrock. Once you've attached, you need to establish, and establish is the process of growing. And in this, for example, we produce a rhizode. Most seaweeds will produce a rhizode, which will turn into a holdfast of the alga, allowing attachment, permanent attachment to a substrate, not a root system. This all comes together in this cycle here that I extracted from an earlier paper by Skeel and Foster in 1986, and that most people are concentrating on the adult plant stages. But what all of these things that they've identified here are largely overlooked in the literature, but equally important for the success of the species over time. Okay, so in the next lecture we'll be looking at some of the adult plant growth, but we'll also be looking at some of these limitations on these individual elements of the life cycle. But a good example of how simple seaweeds can be, but at the same time how complex they are and how dynamic they are. So they are really interesting and really important. And I hope that your interest has been piqued in seaweed and you will have a little bit of a read in the literature that I suggested. I suggest reading chapter 1.5 in Lubbock and Harrison Volume 1, chapter 2 in Volume 2, if you have it, and then doing chapter 5. Looked at some of the morphogenesis life history elements. But Skeel and Foster is the key paper that I want you to look at, because it gives you some excellent examples that you can use to really illustrate how adapted seaweeds are to the environment that they live in. Thank you very much for listening.